Okay, welcome back everyone for the afternoon session of day one of our Revitalizing Biophilosophy Conference. I'm very excited to welcome our next speaker, Professor Alan Rodrigues from the Rockefeller University, where Alan is a research assistant professor and co-directs the Laboratory of Morphogenesis with Dr. Amy Shire. Alan, thanks so much for joining us. And do you need to share your screen or?
Yes, I'll go ahead and do that.
Yeah, floor is yours. I'll give you a 10-minute warning. You got a 45-minute block, and we've been doing questions at the end. So take as much of the 45 as you want.
Okay.
If you want to save some time, I'll give you the 10-minute warning in case you lose track.
Sounds good. Do you just see the slides or do you see everything right now?
We see your notes as well.
Okay, all right, so it's okay. I don't need them. How about that? Is that better?
Yep, that's it.
Okay, great. So yes, thanks, Matt and Spiridon, for hosting this very diverse conference. And yeah, as Matt mentioned, my day job is to co-direct a research laboratory. So I'm in charge of thinking about executing experiments and doing work in sort of mainstream molecular and cellular biology, but at also levels above that. And I think the view that I've come to have over the course of my career is that there's that most of the bottlenecks in laboratory research are actually not of a technical nature. They don't have to do with not having the right tools, but rather they're really of a conceptual nature. And it's really the way that we're thinking about biology that is really rate limiting. And so I think the work that we're trying to do in the lab is very much obviously trying to innovate in technical ways and also to deal with questions that are really not understood at the cutting edge of biology, but to do that in light of a new set of ideas and concepts. And I think if I were to distill it all down, I think that the main issue is that there is really an over-reliance on concepts that really rely on bringing together sort of machine-like views of living systems with the assumption that cause really emanates from the molecular and cell and subcellular scale. And so just to give you an outline, and I should say, I think I think it's very important that laboratory scientists get more exposure and take really seriously conceptual thinking. And I think it's really great to have situations where philosophers and laboratory scientists can speak and relate. So I think this is a great opportunity. But at the same time, it's the first time I've given a talk to a more philosophically oriented audience. So I think I run the risk of being both too technical and also philosophically naive. So I apologize at the outset, and I hope you can set me straight in the Q&A, and I'll try to make sure to leave lots of time for that. So in terms of the outline of my presentation, I'd like to just give you my take on what I think to be the current state of laboratory research in the biological sciences. This is not a prevalent view. There are some people in the audience who would agree with me, I think, who are also laboratory scientists, but I think this is probably a heterodox opinion. But I'll share that with you to sort of give you sort of a sense of where I'm approaching things from. And really having to this idea of there being a crisis, because of this view of sort of life being sort of emanating from the scale of the molecular machine. And then I'd like to present the strategy that Amy and I have been trying to use in our laboratory to try to methodically kind of move out of that paradigm, which kind of exists as sort of like a straitjacket that you have to kind of try to extricate yourself from in a strategic manner. So I sort of explain to you our strategy for that, and then share some specific concrete examples from our laboratory research that have been done in the light of what I would call an organic conception, and then briefly share some implications for biomedicine that we're just dipping our toes into, and a few pioneers have already made arguments for. And then finally, just like very briefly end on just like reflections of method, of what I've sort of come to think about what we're doing in the lab when we try to approach things in this quote unquote organic way. So, right, to start off with the sort of current state of things, I think without being, too dramatic, I think I would say it's a crisis. But I think it really starts from a reliance on machine metaphors to characterize living things. This is not often explicitly stated. It's often sort of just embedded in the language that people are using, or the type of logic that they're using to sort of make their arguments. But you will often have references to things like blueprints, a focus on things like encoding, the idea that cells are sort of making very precise calculations or computations, and then sort of the idea that whatever phenomena that you're studying, in a sense, is under the influence of some sort of external control. So, for instance, if you think about the topic that I'm going to speak about later, morphogenesis, the idea is that the form that you're seeing coming into being at a more macro level is being, in a sense, controlled by some sort of influences at the molecular level. And you can see a combination of these types of metaphors. I wouldn't say that these ideas are brought together in a very rigorous or coherent way, but they are, in some sort of loose, disorganized way, they do sort of undergird the thinking in much of biology. And an example of this is the gene regulatory networks that were developed by Eric Davidson, who's a very influential scientist working out of Caltech for the latter half of the 20th century. And again, this sort of machine perspective is really wedded with a perspective that's rooted in molecular reductionism. So there's sort of this idea, there's a quote here by Jacques Monod, saying that the cell is indeed a machine whereby the microscopic clockwork function that establishes between DNA and protein an entirely one-way relationship. So this idea that, if you can understand the mechanical interactions of what's going on within the cell, you'll really go a long way in understanding many of the phenomena in life. And that's really been the focus of more and more mainstream biology, even when what is of interest could be a phenomenon that is far larger than the scale of a cell, like a tumor, for instance, or a developing organ. And you don't need to have a PhD in the biological sciences to wonder if there are limitations to this, right? I mean, I would say the two main examples here is that a machine doesn't really have an intrinsic capacity to organize itself. It doesn't build itself, and it also can't necessarily repair itself if it's damaged in some way. And this is an example of phenomena that are sort of very apparent in living systems that sometimes get sort of pushed to the side when you start to focus in this very kind of reductionist way. It also has a very practical limitation, which is that if these paradigms are sort of inaccurate, it's one thing for it to just produce papers of poor quality in the academy, but we may be sort of undertaking medical and sort of therapeutic approaches that aren't really necessarily going to lead to a lot of beneficial outcomes. So, for example, if diseases are not merely the consequence of codes embedded in the DNA, then the billions of dollars that we've spent on gene sequencing may not actually pay off and result in some of the promises that were made. And there's this term of paradigm instability. So we've sort of this idea of things emanating from the level of a molecular machine in order to account for a lot of the anomalies that have sort of arisen has just sort of gotten more and more complicated, and there's sort of been additional patches sort of put onto it. And some people have argued that we're reaching this phase now where it's in a sense almost caving in on itself. And some people would argue that is showcased in our inability to understand and treat diseases like chronic diseases and things like cancer. So, and this is also starting to be acknowledged now by leaders in the field. The current FDA commissioner, admits and even acknowledges. I mean, it's in a sense a critique of the paradigm that he maybe wasn't a part of, but sort of critiquing the idea that, in a sense, the gene is at the root of all of our problems. So, now I just wanted to just briefly talk about sort of our strategy to kind of move beyond this paradigm. And I will say it's not merely just an intellectual issue. It is also a sociological issue with sort of the structure of academia and the larger context within which academia sits. So you may have a paradigm that intellectually one could argue with and individuals could disagree with, but it's really embedded in a larger sort of reinforcing kind of network of causes where you have more technology dedicated just to that level of understanding. You have for-profit interests that are sort of providing additional resources to sort of keep things in a particular view because it may serve their interests for developing therapeutics. Under the view that if one understood quote unquote molecular control mechanisms, then you could devise ways to manipulate them. And then there's also sort of a feedback there with how public resources are allocated, and also things like prestige and awards. So it's not merely an intellectual issue. It's also a sociological one that also I think also in a sense keeps things where they are. And so it's not trivial as an independent, as a scientist within the system to think about how to move beyond it. Our approach is to really shine light on the phenomena that the sort of molecular machine view really struggles to explain. And for us, the key phenomenon, which in a way is to me at least, I'm biased, but to me it's the phenomenon of life is how form emerges. And for the purposes of this talk, it's really about how form emerges in vertebrate organs. So it's a situation where you're forming these morphological structures that are comprised of many, many, many cells, right? So it's not the difference in length scale between a protein and the thing that you're interested in is quite large. And what's interesting is we at this point have an extensive catalog of the molecular parts that are present. We have the capacity to delete any gene at will now with CRISPR technology. We have the ability to put in any genetic material we want into these systems, and yet we still don't have a clear understanding of how form emerges. And so I think we sort of think it's a nice place to sort of move beyond the status quo because I think the status quo has really struggled to deal with it. And I would say that the general idea here is to, in a sense, take a morphocentric view rather than a genocentric view. So the idea is that, in a sense, form or organization is the principal phenomenon that you anchor to, rather than the sort of genetic or molecular changes and the amounts of various genes, for instance, that could be being transcribed from the genome. One other key sort of ingredient is sort of to take this, what one could call a post-molecular perspective, which is, to on the one hand acknowledge that we've learned a lot from cataloging and understanding the sort of networks of biochemistry within the cell, but that there are other scales beyond the individual cell that need to be characterized. And moreover, that these other scales may in fact actually give more meaning and understanding to the characterization that we've achieved at the subcellular level. And the idea there is to use, in a sense, physical concepts, not to drill down into smaller and smaller pieces, but to actually try to recover aspects of the whole and give them some amount of rigor. So, the scale that we're very interested in is, what one could call the supracellular scale. So it's the scale that's larger than an individual cell, but smaller than an entire organ. And if you look at most textbooks or just, diagrams of biology, this sort of intermediate scale is one that is rarely really thought about, or it's usually missing. And if one thinks about how things are often communicated in biology, there's sort of an appreciation of obviously the organ level. But then you very quickly move down into the sort of molecular and cellular level when one wants to, quote unquote, understand the causes of what's happening. Now, the other thing I will also mention is, this is another feature that I think is very hard and I think has really been sidestepped by the kind of molecular machine view, is the phenomenon of self-organization. So I think this is the other thing that we really try to shine light on, which is to try to identify key instances of self-organization that we then would like to argue are really central to the phenomena that we care about, which in this case is achieving structure. And I would say 10 years ago, I think it was very much the dominant view was very much that form is really controlled through some sort of a molecular process. And I think more and more there has been a shift into just acknowledging that there's also something self-organizing happening that in a sense is emergent in terms of form. And this is an example here where we have basically a collective of 20,000 cells that are arrayed in a ring. And then when we plate them, these are taken from the embryo. These are dermal fibroblasts, and we scramble them up and just place them next to each other. 20,000 of them onto a sheet of collagen. And over the course of about 2 days, they, in a sense, self-organize into these aggregates that are, roughly the size and spacing of the follicle aggregates you see in the developing skin. And in a sense, what an assay like that in our work is suggesting is it's not merely those aggregates that we see in the embryo are not just the consequence of molecules telling the cells where to go. Because when we put those cells sort of arrayed in a ring and sort of leave them to their own devices, they can somehow form these aggregates through their own behavior. And so it sort of calls into question these sort of classic diagrams that have really guided the field of not just development, but also evo-devo, which in a sense assumes that the patterning decision is sort of upstream, you could say, of the cells actually moving and organizing. So the basic evo-devo models is that you actually have these molecular patterns that get formed first, and then the cells and the tissue as a whole kind of respond as a result. And in a sense, by zooming out, I think we're really sort of questioning this entire way of organizing this particular paradigm. So in terms of our laboratory work, and I guess we'd say in light of an organic conception, what do I mean by that? I would say, again, it's this focus on seeing self-organization, or in a sense, intrinsic order creation as being the central phenomena, not just something that needs to be tamed or sort of controlled by a molecular process. To focus on reciprocal relationships rather than sort of linear relationships. So not gene A affects gene B, affects gene C, but rather thinking about when two elements are really simultaneously influencing each other. And so it really becomes not about a search for a thing like a master gene, but really about identifying the right reciprocal relationship in space and time and associating that with the phenomenon you care about. And then to appreciate the existence of emergent properties that are associated with higher levels of organization and the fact that there can be top-down causality. So it's not merely just sort of genetic and molecular information flowing upwards, but that can actually go in the other direction as well. And so, if you take that all together here, the idea is, people often think about, this nature-nurture divide, right? Where you have your genes coming together with your environment. And so the idea is like, well, if you can, in a sense, add those together and understand what happens to the person. But that's complicated if there are actually self-organizing systems at levels beyond the gene, but still within the organism. And in a sense, we're trying to uncover that in concrete instances to explain open questions in morphogenesis. So this is just the embryonic context for that ring experiment I showed you. This is for what happens with the embryo. So at day 6, this is an aerial view of the bird of a chicken embryo, day 6 and day 8 on the right. And you're looking at the back of the embryo. And in 2 days, the back becomes studded with these follicle aggregates. And you can simplify that process a little bit because these aggregates form in rows. And in a sense, our ring is, in a sense, a simplification. It's a reconstitution of the organization of that strip down the middle. And so the fact that we were able to take these cells out and put them in the system, I think, is an argument for the fact that these cells are collectively self-organizing and not merely being instructed by a molecular pre-pattern. But then we wanted to kind of come up with an explanation of, well, what is happening here to account for this self-organization? And the key, one of the key things that we sort of came to realize is that it really has to do with the reciprocal relationship between the cells and the matrix that they're in. And so there's really a dynamic relationship where the cells are contractile and mechanically active, and they reorganize the matrix. And as that matrix gets reorganized, it sort of guides and causes the cells to become more and more aligned. Almost that's akin to a pneumatic state that people often identify in physical systems. And so there's this reciprocal relationship, and this takes time. So this is what's happening in the course of, let's say, 12 to 16 hours. And so that's a dynamic process. And we saw a similar process happening in vivo when we looked inside the embryo. And then what was also important to realize for us is that the interconnected tissue, when the cells become aligned in the cell ECM network, and in a sense are reminiscent of what people refer to as liquid crystals, the whole tissue, in a sense, can adopt a set of properties that are akin to a fluid. And so if you think about, so you're really thinking about things at 2 scales. You're thinking about the cells in the matrix interacting at a granular level, but then at a larger level of organization. That granular level sort of leads to an emergent property of you could call supracellular fluidity. And if you think about water, something like water, you've probably seen beads of water beat up on a leaf. Or here's an example, right, where if you streak some water in a line, or in this case it's a circle, over time it can sort of aggregate into spontaneously aggregate into aggregates. And in a sense, what we did through some theory and experimental work is to argue that at the supracellular level, the collective of cells behave like a contractile fluid. And the key thing here is that that emergent property is what just directly organizes the follicles, right? So it's not like the genes within the cells, or sorry, the individual cells need to be told what to do by a chemical cue. It's actually, so it's not a micro sort of decision. It's actually a macro property that, in a sense, is guiding the entire aggregate of cells to behave in this way. That macro property is dependent on, obviously, the features of the cells and the matrix, but what's guiding it is really the whole rather than sort of the individual parts marching into a place because of their being told to go there by a molecule. So, what I just spoke about is a concrete example of one particular study that we published a few years ago now. One thing I'll say here is that what is interesting is that there is, when we combine that insight with insights from other studies that we've done, it's not merely that sort of this supracellular or emergent property can organize the aggregates, but as those aggregates are forming, they can actually compress neighboring tissues. And that neighboring tissue that becomes compressed, the individual cells in that tissue can sense that compression and then activate a whole new set of genes within each cell. And so there's sort of an inversion of causality, not from the micro to the macro, like that. When I went back to the oh, sorry. it's an inversion of this type of causality here. In this case, you're really having the molecular patterns appear first and then guide the higher level morphology into a pattern. What's happening um, what's happening here is that it's actually the collective of cells that are deforming a neighboring tissue, which then sort of change the genes within it. So there's sort of a reversion of that causality. So this is we wanted to start our work in a very sort of fundamental area where there was a lot known, where people thought they sort of understood the whole system and sort of challenged sort of a certain entrenched paradigms. But I don't think it takes a lot to see that there are implications for biomedicine. So I want to just speak very briefly about thinking about disease and how we think about disease. So this is an example of a diagram from a very well-known review by Hanahan and Weinberg. It's a highly, highly cited review. A lot of cancer papers cite this review. And you don't have to really focus on the details, but the key thing to notice here is that every single cause that they're associating with the sort of onset of cancer is in a sense subcellular. They're all subcellular events. And so it's a very molecular-centric view. There are some noticeable exceptions to this perspective, but I would say this really dominates the practice of cancer biology research. But I think what we are thinking about in general is the idea that a disease state, rather than just in a sense being the cause of molecular mutations or changes at just the molecular level, are also to some extent a manifestation of self-organizing processes at levels above individual cells. And again, you could think about with cells in matrix, issues with the stroma and things like that, where you really have dynamics that are happening at that length scale that you really that may feed back down on the molecular level, and certainly can't just merely be reduced to single cell characterization. I think in addition to cancer, I think it could be a way to also be thinking about chronic disease. So there's certain hallmarks of chronic disease having to do with fibrosis and inflammation and metabolic dysfunction. And in a sense, to view it, to sort of take, instead of a genocentric or a molecular-centric approach, just to take, in a sense, a morphocentric approach, which is just to see it as a structural, or in a sense, a pathology of form, rather than the amount of a particular gene. And in a sense, that morphological pathology, you could say, could be in a sense, the central organizing causal nexus for things like inflammation and pathologies of cell metabolism and fibrosis. So that's sort of the way we're thinking about things in disease areas, and again, just sort of trying to acknowledge and appreciate this level of organization that in a sense has its own agency, you could say, because there's a self-organizing capacity at this length scale, and that cause may be distributed, beyond just the cellular level. This again might seem obvious, but it really is stunning to see how little research in disease it really acknowledges levels beyond the cell. And then the second thing is, but in order to acknowledge it in a new way, I think one really needs to employ a different set of conceptual frameworks that come out of work by sort of this organicist lineage that's been underrepresented over the last century, which I think is also very critical. So now I'm just going to end on something that I think is more of a reflection and not something that I could really prove, but it's just something that has come more and more to my mind as I've just thought about how we teach our method of doing science to students in the lab and more broadly. And I've just come to get the sense that a lot of the key insights in our lab, or the key advances, were not made merely through the development of a new tool, but really came about through meeting what we saw empirically with an intuition that came from us. So there was really something of a coming together of you could say a percept and a concept that was coming from us that in a sense revealed the fuller or truer reality of what was going on. And this capacity, this, I don't know what to call it in a rigorous way, but this intuitive capacity that is necessary to complement what you see empirically, I think is very essential to doing organicist work properly. I can see how you don't need it as much if you're just mechanically reducing things. But if you want to do things and really deal with the fluidity of life in a very active way, you need this form of living intuitive thinking. And it tends to get de-emphasized in a culture that focuses on technical mastery of technology, and now more and more the use of AI to, in a sense, capture nonlinear interactions in a model that's hard to understand and somewhat opaque, as a way to sort of get around the fact that sort of a human being could also potentially, through their observations, come up with an idea of what is happening. So that's just, again, I don't know if it's something I can prove. There was an article written by Frederick Amrin on Goethe and emergence, which for me was very clarifying. But I do think it's in some ways even more rate limiting than an interest in conceptual thinking in laboratory research. So with that, thank you for your attention, and thanks again to the organizers for giving me an opportunity to present.
Thank you, Alan. Yeah, lovely to have the laboratory perspective. It confirms for me that in the criticisms I'll share in my talk of the dominance still of the mechanical metaphor in biology, that I'm not just making that up. It's a real issue. Look at all those questions. All right. Josh, your hand is first. Make sure you unmute. There you go.
Yeah, thanks.
Oh,
Yeah, that
one
was
sec.
great. Oh, sure.
Alan, can you just stop sharing your screen?
Oh, yeah.
Thank you.
Um, yeah, that's great. Thank you. I really like how the way you characterize the top down, the simultaneously bottom up, top down, is an ongoing question that we're all really interested in, and I really liked how your work characterizes that. My question is, in terms of like specifically your example of cells, the genetic network being, modified or affected, if you will, by more distributed notions of pressure, for example, or tension, or any different mechanical, more distributed mechanical phenomenon. Have you guys looked into how it is that sensitization, that capacity for the operation of the gene, the genotype to the phenotype, like how it is that emerges, how that sensitivity emerges in the system, like how novel sensitivities could potentially emerge?
In relation to top down, or?
Well, no, actually both, like simultaneously, as you've nicely characterized how it is the system, if we understand it, is simultaneously bottom up, top down. That there's process in that sense, in the sense that there's a dynamic whereby the gene expression is affected by, say, pressure, as you illustrated. But how is it that the system as a whole, both bottom up, top down, becomes sensitive to pressure in the first place? Like how it is sensitive in the sense that the gene expressions will be modified.
Yes. So specifically in this case, there are proteins that are attached to the cell membrane normally, and when the cells get compressed, there's a conformational change in the structure of the membrane, and then that protein no longer binds to the cell membrane, and that protein also has an affinity for the nucleus. So this is a beta-catenin is the compound. So basically, it's a protein that can attach to the membrane or DNA,
Mm.
And in a particular conformation, it preferentially binds to the membrane. But when the cell is manipulated and changes conformation, its affinity then gets shifted to wanting to bind to DNA. And so that's an example. So they're all, I mean, it's called mechanosensation. It's this whole new emerging field where, in a sense, there are all these cells that can sense cytoskeletal changes, the change of the cellular structure, and as the cell structure changes, can basically change their affinity and activate a signaling cascade. Most people tend to just enjoy characterizing the transmission of the signal from that sensor to the nucleus. But I think the key meaning of it is to enable top-down causation.
Yeah, excellent.
That's what's exciting. I think we're really interested in finding all of the kind of supercellular impulses from larger length scales down to that level.
Great, thank you.
Thanks, Josh. Alfredo, you're up.
Thank you. Great presentation, Alan. Thank you very much. Another way to understand your presentation is the power of metaphors. You're talking about a lot of metaphors in the work. I am a very fan of Ronald Latour's work, Our Laboratory Life, from 1979. And Latour, in this book, he writes about how we can construct scientific data, how we construct facts in science, and metaphors are part of this work. So in your last slide, you also talk about intuition. So my question is, how do you document, how do you register the metaphors, the intuitions? Because it's not so easy. It needs another level of interpretation. So my question is, do you have a documentation about this metaphor thinking?
Yeah, and when you say document, you mean bring out of your own mind? What do you mean when you say document, or do you mean
writing?
Or how do we write about these metaphors? You're saying?
Yes.
Well, it just so in one way that we do it is that I mean, I don't know if this quite gets at your point. I think one issue that we deal with every time we try to get a paper published is that we are using a different set of, we are using the same words, but in a different way. So for instance, like the term epigenetic has become reduced to just thinking about chromatin marks. But the original conception of that word was far broader. So there's a battle in stretching these words, I think, in a pragmatic sense, when you're going through the review process with your peers, many of whom think very mechanically. I don't think I don't think I have a systemic explanation. I think one thing that we've done, I think we use biophysics in a way, basically. I think the use of physical concepts allows us to make things like emergence, which some people would say sounds like hocus pocus, like a very real thing. You know, like we've done experiments where we will invoke these concepts. I don't know if I'm quite answering your question though.
But I mean, the work in the laboratory also changes your understanding of the concepts.
Yes.
So this process changes also the metaphors.
Yeah.
They change it from the beginning to the end of the process. So it's like to think about a journaling about metaphor thinking.
Yeah.
Do you have something about this?
In the lab, no, but I mean, I think one thing that it drives people in our lab sometimes a little bit, frustrates them, is that the understanding of our project changes in the course of the project. So it's not like it's fixed. Mm-hmm. You know, someone may want to know what their project is when they start it, and I think there is an evolution in our understanding, even of things like causality, right? You know, understanding, you know, something that we're thinking about right now, which is changing. It feels like it changes day to day. It seems to me like there's something very different there's a deep difference between bottom-up and top-down causality. They're not symmetric. And so we think about that, but we are constrained by what we can see experimentally too. So I think that's the other thing. We're experimentally also limited. But I guess to say it broadly, it's a good question. I don't think we have figured out how to systematize that. But it is an issue, certainly.
Okay, thank you.
Thanks, Alfredo. Paul, please.
Kia ora, Alan. That was really wonderful. Look, I suppose I've got so many questions from this, but I'm not gonna. I won't dwell on them too much. Like, just to give you a little bit of background, I'm a physiotherapist or a physical therapist, and so working in pain science really. And my thesis, my PhD, which I'm currently embarking on, is looking at an ontology of pain. So seeing if we can try and think of pain in a very different way. But really, I was just wanting to make a comment about the start of your presentation, where you were talking about machine metaphors and such like. And it kind of gave me a parallel with regards to how we practice in pain science at the moment. For example, there are things called mechanistic descriptors. I mean, just the word mechanistic descriptors to classify or categorize the type of experience someone will have, and a pain experience someone will have. So, for example, there's a mechanistic descriptor called nociplastic pain, which is now, it refers to the nervous system, a change in the way that the mechanisms of the nervous system or the machine body works that gives rise to the experience of pain. And what's really interesting about your talk is that you talked about form. And so the challenge I have is that when I look at form in your pictures, there are different colors, there are different qualia that I see within that. And I'm going, okay, I can kind of get a sense of that. But when it comes to something like pain, pain doesn't, in my mind, pain doesn't it doesn't have any kind of form except for how we might put it in this kind of mechanistic way. And so that was really just a comment, because it just got my mind thinking about that stuff that you were talking about there. And then just really a question I had for you specifically. You talked about mechanosensation. There's a phenomenon that I'm aware of in the physiotherapy world, in the biology world, called mechanotransduction. And my understanding is that mechanotransduction occurs as a result of the molecular exchange that goes on within the cells. But from what you're saying, if I've understood, is you're saying that there's this kind of almost kind of inherent, and apologies if that's the wrong word, inherent self-organization that occurs, that's the whole. So it's not the molecular part to the whole, it's the whole itself. I mean, that, if I can be crude, that sounds, from what I've been reading around Bergson, that sounds like there's some form of memory that's going on in there.
Yeah, I mean, I think there's a lot going on with self-organization that we may not be able to capture with just material biophysics. To your first point, I will just say that, I think that the idea of pain, I think of pain as something very different than the capacity to grow and form something. In a sense, like, there's a difference between like sort of a formative force of growth,
right?
Which I think can be somewhat captured by characterizing fluid temporal processes. But I think of pain as some almost having like a very different dimension of quality than purely growth. So I don't know if I can say something specifically about pain, although I certainly think that those types of sensations are somehow correlated with morphology. I certainly think that, but I think I'm sure there's a correlation between pain and form, but it's different than the formative forces that I think I'm talking about here.
Yeah, no, makes sense. Yeah, yeah.
Yeah.
Thanks, Paul. Let's try to get one more question in and then we'll transition. Manfred, you're next. And for those of you who still have your hands up, drop it in the chat if you want and we'll continue there, but we have so much time. Go ahead, Manfred.
Yeah, I have a short question. So since you are at Rockefeller University and into developmental biology, I was wondering whether Paul Weiss still plays a role in research there, because he was an eminent figure until the 1980s.
Yeah, I guess I didn't. Yeah, I think he's been in my promotion talk. I ended with a quote by Paul Weiss.
Okay.
Yes, but it's well, he passed away. I think in the 80s.
Yeah,
it's been a while since he passed away. I think it's a little bit sad, but I think if you poll, I would say most of the faculty wouldn't know who he is today at Rockefeller. I think it just speaks to the grip that molecular biology has in mainstream biology, but I don't know if any of them would. Maybe some of the very, very senior ones, but no one under the age of 50 certainly would know who he is. So that would give you a sense of how influential he is here today.
That's too bad.
But we are trying to revive. I mean, that's what Amy and I are trying to do. I mean, I think we're trying to revive that. And there are people. I mean, people like Carlos and Anna who are in the audience who I think have been bravely carrying that torch.
Yeah, because it was also talking about macro determination, things like that. What you were talking about.
Yeah. They just didn't have, they didn't know about mechanotransduction then, right? Or they didn't have, like, I think now you can actually go back and with, and that's what I use this word post-molecular. I think we can use a lot of these features that we can now characterize to actually give more specificity to the concepts that people like Weiss were arguing for.
Great. Thank you, Alan.
Thank you.
So we're going to move along here. Let me switch.